Good morning, everybody, or good afternoon. It is a pleasure for me today to present Shelly C. Shelly is professor of medicine at the Keck School of Medicine in the University of Southern California, and associate director of the NIH-funded USC Research Center for Liver Disease, and associate chief in the Division of Gastrointestinal and Liver Disease. She got an MD degree from UCLA, and then did her internal medicine there, the residency, and then became also assistant professor at UCLA, and then moved to University of Southern California as a professor around the 90s. So she has done all her career in Los Angeles, which is a little bit unusual for an American to stay all the professional life in one place. She started working on liver and glutathione, was trained by Neil Kaplovich, who you may remember. He was here giving a seminar not that long ago, and then developed a very successful independent career in liver disease. We met first time, I think, was in the early 90s. Ever since we have been collaborating, we share a number of common interests. And she's been funded by NIH since also the early 90s, 91, 92, which, and at present, if I remember well, she has 4 NIH grants, which is well, tells about the quality of her work. Also, I think tells about the quality of her work that in January, she received the WSCI Mayo Solly Award for Lifetime Achievement in Science Mentoring, and is the first woman to get it, and is also the younger person to get it. Interestingly, her mentor to research also got it, but only a few years ago. So I think you are ahead about 10, 20 years of Neil. So anyway, it's a pleasure to have you here. And the title of her lecture is, you can all see, What's New About MAT Gene and SAMI, a subject which is close to some of us. Thank you, Shelly.
It's really been just a pure delight, really, to have heard all the wonderful work that's going on here. We had a marathon session yesterday, and it was wonderful. And it's been a great collaboration. Jose is one of my closest friends, really, in the world. And it's been such a joy to have known him and work with him, and many of you here as well. And I don't think I need to tell you guys what MAT genes and SAMI are, since your director really is the world's renowned expert leader in this field. So I will share with you some, I know some of you were in the lecture yesterday, so I apologize. Some of the background material will be repeated, but I think many of you were not there, so I will reiterate some of the stuff again. And if you're like me, I can't remember what I learned yesterday, so. All right, so I know that y'all love this pathway, so is there anyone that doesn't know this pathway? Otherwise, I can just skip this pathway, skip this slide altogether. But just in case, all right? So now this is our molecule of interest, S-adenosylmethionine. So this is the pathway that illustrates how the liver metabolizes SAMI. So liver is where half of the daily intake of methionine, which is an essential amino acid, is catabolized. So the catabolism of methionine is via its conversion to SAMI by this enzyme MAT, methionine adenosyltransferase. Now, SAMI in the liver, in the hepatocytes, participates in 3 very essential reactions, pathways, I should say. One is in polyamine biosynthesis, and this occurs in all mammalian cells as well, not just the liver, where SAMI is a precursor for these polyamines. okay? And in the generation of these polyamines, SAMe give rise to MTA, methylthioadenosine. In addition, SAMe can be spontaneously converted to MTA. So oftentimes when we're talking about treatment with SAMe, you want to keep in mind that MTA may be playing a role. SAMe is also the precursor for glutathione, which is an essential antioxidant through this pathway called transsulfuration, where homocysteine is converted to cysteine, which is the rate-limiting precursor for glutathione. And as you all know, SAMe is the methyl donor. So the transmethylation reactions all require the participation of this donor. And in liver cells, the quantitatively most important methyltransferase that many of you already know is glycine N-methyltransferase. It accounts for 1% of the cytosolic protein, okay? Now, in hepatocytes, homocysteine is regenerated back to methionine by both betaine homocysteine methyltransferase and methionine synthase. Now, methionine synthase, as you can see, is now closely linked to this folic acid cycle, and it requires presence of folate, B12 for this cycle to work. Other vitamins are required in this pathway, like B6 for transsulfuration, okay? So this is SAMe, and it participates in those pathways, as I just illustrated. All right, now let's go on to MAT genes. I know you saw this slide yesterday, many of you. So in mammals, there are 3 genes, MAT1A and MAT2A, encode for the catalytic subunits of MAT, alpha-1 and alpha-2. Alpha-1 then form a dimer or a tetramer, and then alpha-2 also forms a polymeric structure, but it varies depending on the tissue you're looking at. And then there is a regulatory subunit encoded by MAT2B. This beta subunit lowers the Km of MAT2 for methionine, but it also lowers the Ki of MAT2 for the product SAMe. So by doing that, it makes MAT2 function more efficiently at lower substrate concentration, but it prevents the SAMe level from fluctuating too much because of this feedback inhibition, okay? The beta subunit does not regulate MAT1 or MAT3 that we know of. Now, in fetal liver, this is illustrating where they're expressed. In fetal liver before birth, MAT1A is not present. Liver has MAT2A and MAT2B in that stage. A few days after birth, there's a switchover so that MAT1 is induced transcriptionally, MAT2 becomes silenced, and in the adult liver, the hepatocytes, which accounts for the bulk of the liver, okay, really does not express MAT2A or MAT2B. It's just MAT1A. But the liver has extraparenchymal cells, such as Kupffer cells, endothelial cells, and stellate cells, and they express MAT2A and MAT2B. Now, the situation in liver cancer reverts back to the fetal phenotype, so that most liver cancer, MAT1A is either downregulated or altogether absent. Then you have induction of MAT2A and MAT2B. Okay? MAT2A also gets induced whenever the liver is growing rapidly, such as during regeneration following a partial hepatectomy, when you feed the animals a carcinogen, and even with ethanol feeding, there's induction of MAT2A. And also, actually, unpublished, MAT2B is induced with ethanol feeding in the animals as well. MAT2B, that was a work of Malu, also showed that in human cirrhotic livers, MAT2B mRNA level is increased. Okay? Outside of the liver, MAT1A is largely absent if you use like a northern blot. If you use RT-PCR, very sensitive method, you probably would see it in a lot of tissues. But the one tissue where you would see it abundantly is pancreatic acinar cells. Okay? Pancreatic acinar cells and the liver share embryologic origin, and that may explain the reason why. Now, MAT2A is expressed in all extrahepatic tissues. But MAT2B is expressed in most, but not all tissues. Okay. Now, even though these enzymes, MAT1, 3, and 2, all catalyze the formation of SAMe, these isoenzymes have very different kinetic properties, and they are subject to feedback inhibition by SAMe very differently. Okay? First of all, SAMe inhibits MAT2, doesn't do it to MAT1 or MAT3. SAMe inhibits in the pre-translational level the expression of MAT2A, but SAMe maintains the expression of MAT1A. So depending on what MAT is expressed, you know that your SAMe level will be quite different. Generally, a cell that expresses MAT1A, you will have high SAMe level, especially if you feed it with a lot of methionine, okay? But a cell that expresses MAT2A, the SAMe level will not accumulate much because also of this feedback inhibition, all right? Now, it's been known for a long time, largely work from Jose many years ago now, that patients with cirrhotic livers, this is a picture of cirrhotic liver, there's low MAT activity. It's reduced by about half, okay? Now, we then show that actually it doesn't have to be end-stage cirrhosis. Even patients hospitalized with alcoholic hepatitis, some of whom did not have cirrhosis, only fibrosis, there's already low MAT expression, no, MAT1A I should say, and SAMe level was reduced by more than half, okay? How does it occur? Well, here I'm showing you the mechanisms. One is pre-translational. This is work, again, from Jose many years ago, and that they show cirrhotic livers, about a third of them, there is no detectable mRNA, MAT1A mRNA, okay? And another half, it's reduced. Now, whatever isoenzymes is still getting made gets inactivated. So there is a cysteine residue at 121. This is only present in the MAT1 and 3, not in MAT2. The cysteine 121 is a target of covalent modification. Covalent modification by nitric oxide and hydroxyl radicals. And when it is covalently modified, it loses activity, okay? Now, you can reactivate this with glutathione, but patients with liver disease often are malnourished, and there are other reasons for them to have low glutathione level. And I already told you they have low expression of MAT1A at the mRNA level. So you couple that with a low glutathione level in presence of oxidative stress, nitrosative stress, you can envision that you run into a vicious cycle, okay? And that is why they have low glutathione level and they have low SAMe in patients with liver disease. All right, so more than a decade ago, doesn't seem that long ago, I guess we're old. Anyway, so Jose and I decided to collaborate and create the MAT1A knockout mouse model. This is a model that would allow us to address what are the biological consequences of having chronic hepatic SAMe deficiency. And this would mimic what happens in most patients with chronic liver disease, okay? So in the absence of MAT1A, there's induction of MAT2A. And this is necessary because, after all, we have to have SAMe, okay? There has to be a source of SAMe. Otherwise, there's no methyl donor, okay? So MAT2A expression is induced, as is alpha-fetoprotein that's induced. Alpha-fetoprotein, for those of you not working in liver, is a marker for dedifferentiation and rapid growth. In fact, in hepatology, when you treat human liver disease, this is something that we check every 6 months in patients that are predisposed to liver cancer, okay? When it goes up, we worry about liver cancer. All right? Now, plasma methionine level went up more than 3-fold. So this illustrates how important MAT1A encoded enzymes are in handling a systemic methionine load. And in fact, if you think about the enzyme kinetic, it makes sense that you have to have MAT1A encoding for those 2 isoenzymes that don't get inhibited by SAMe to allow that organ to clear methionine. Okay? So if you get rid of it, the plasma methionine level climbs. And indeed, we see that in patients with alcoholic liver disease, is that they have hypermethionemia, all right? Now, in the liver, SAMe level, of course, fell, and that's just consistent with what I have already told you about their enzymes, kinetics, and feedback inhibition properties. And glutathione level fell, and that illustrates how important SAMe is as a precursor of glutathione. In fact, that transsulfuration pathway that I showed you on the first slide is particularly active in hepatocytes, but not terribly active in most other tissues, okay? Now, the phenotype of this model published more than 10 years ago now, the mice are totally viable because after all, MAT1A is not expressed until a few days after birth. And by 3 months, they look completely healthy, except the livers are bigger, okay? And histologically, the liver looks normal. But if you were to feed these mice a choline-deficient diet for only 6 days, nothing would happen to the wild type, but the knockout mice would develop massive fatty liver, okay? And by 8 months, on a normal diet, they develop histologically steatohepatitis. So for those of you not accustomed to looking liver histologies, this is normal liver hepatocytes, and these open, round spaces, that's fat, okay? So those are fat. And this, in dark dots, those are inflammatory cells. So this is inflammation and fat, what we call steatohepatitis. For human liver disease currently, fatty liver is probably the #1. It's an epidemic, okay? And 3% of the US population is believed to have this, NASH. And this can progress to cirrhosis and then predispose to hepatocellular carcinoma, okay? And indeed, by 18 months, majority of these mice develop hepatocellular carcinoma, okay? All right, so this model is great. It's got a phenotype, so we were delighted, and then, which is cancer and NASH. So using this model, we've been busy in the past few years trying to understand what are the molecular mechanisms that can predispose these mice to develop liver cancer, okay? And the first is that they have cancer stem cells when they're old, okay? Now, for those of you working in cancer, you know that cancer stem cells is increasingly more important in oncology. And in liver cancer, it's believed that about half, up to half of liver cancer originated from cancer stem cells. Why do we think that? Because liver cancer is often heterogeneous. You open up the tumor, you see very heterogeneous cell types, okay? Why is cancer stem cells so important? Because they are far more aggressive and they are much tougher to treat, very chemo-resistant. So it's really important to understand the origin of these stem cells, how they can be regulated, how can they be treated. Now, in the first project that we started in collaboration with Bart Roundtree, who's now in Pittsburgh, we looked at whether or not, as they age, there's any change in the progenitor cell population. And indeed, there was an expansion of progenitor cells, and that some of these progenitor cells, those that were staining 49F positive and 133 positive, were tumorigenic, okay? So CD133, for those of you familiar, you know this is a cancer stem cell marker, okay? So these cells were tumorigenic. This is the first example that I'm aware where we demonstrated there's cancer stem cells present as the mice age without knocking in an oncogene, knocking out a tumor suppressor gene, or feeding any carcinogen, okay? These mice were just on a normal diet. Now, in addition to being tumorigenic, Bart then went on to show that these stem cells are resistant to TGF-beta mediated apoptosis, okay? Another abnormality that's known to be important. Another pathway that's abnormal has to do with genomic instability. Genomic instability, as you know, occurs in cancer, correlates with cancer stage, and you also know that we have many enzymes that participate to prevent genomic instability from occurring, to fix these DNA lesions. And one such enzyme is APEX1. And in this work, Laura was a graduate student in my lab at the time, Laura Tomasi. She found that APEX1 protein was markedly downregulated in the knockout mice, okay? And there was increased genomic instability, and she showed that in culture, APEX1 protein level rapidly disappeared. But this can be prevented if you put SAMe in the culture medium. So this was one of the suggestions, among others, that SAMe can stabilize protein, probably has an effect on protein degradation, which I'll show you later, okay? Another area that we found to be abnormal, that I'll tell you in a little bit more detail, has to do with uncontrolled ERK activation. That many of you know is a very, very commonly observed phenotype in HCC, in liver cancer, as well as in many other cancers, that you have total uncontrolled ERK activation. And another pathway that is close to Malu's heart is LKB1 and AMPK. This is an interesting pathway because LKB1 is often thought to be a tumor suppressor in other cell types, but they demonstrate that in hepatocytes, they seem to behave in the opposite, so that these mice have increased LKB1 activity, increased AMPK activity, and when this is activated, it led to a nuclear to cytoplasmic translocation of HER. And this is a protein that stabilizes many mRNAs by binding to the 3' messenger RNA end, and many of its targets are cyclins, okay? And this is another mechanism favoring malignant degeneration and growth. And yet another abnormality that I will tell you in a little bit more detail, that some of you I know are working on now, has to do with prohibitin 1, which is a very exciting area that we hope to get funded soon. All right, now this is the DUPS1 story that I'll share with you. So you know that this pathway, heightened ROS activation, ERK activation, increased proliferation, angiogenesis is a hallmark of cancer, okay? Now, ERK and DUPS1 stands for dual specificity phosphatase 1. These two exert a reciprocal regulation against each other. When ERK is transiently activated, it will phosphorylate DUPS1. And when DUPS1 is phosphorylated, it gets activated and turns off ERK, okay? So transient activation turns on DUPS1 to inactivate ERK. But if ERK is activated and this is sustained, so when you have prolonged ERK activation, something else happens. It recruits other proteins and it phosphorylates DUPS1 at a different site. When that particular site is phosphorylated, it sends a signal to send it to ubiquitination, followed by proteasomal degradation. So depending on whether or not you have transient or sustained ERK activation, when it's transient, it's controllable. If it's sustained, it'll turn this off by leading it to degradation, okay? Now, downstream of well, I should say upstream of DUPS1 is P53. P53 is known to transcriptionally activate DUPS1. P53 in turn is downstream of APEX1, okay? Since we already knew from Laura's previous work that APEX1 is downregulated in the MAT1A knockout mice, we therefore suspect that there may be something wrong with DUPS1 as well. And we also knew from early in our work that MAT1A knockout mice livers have high ERK activity, okay? So we already knew going in that ERK was activated and that we knew APEX1 was downregulated. So by this time, Laura was a postdoc in my lab, and I was very fortunate that she decided to stay in the lab and continue working. And so here she shows DUPS1 mRNA level is lower in the MAT1A knockout, and as is the protein. The protein level was fell to even greater extent as compared to the mRNA level. And ERK activity, as indicated by phospho-ERK, is markedly increased in the knockout, and we knew that beforehand. And here is phospho-DUPS1. This is the phosphorylated form that correlates with degradation. Okay, so it's phosphorylated, a different residue. So this is also increased, and this is just densitometry showing that. Now, she then resorted to mouse hepatocyte model. Because mouse hepatocyte, when you put in primary culture, and the same is true with any primary hepatocytes in culture, they rapidly dedifferentiate. And one of the things they will lose is MAT1A expression, okay? Because MAT1A is really a phenotype of differentiated hepatocytes. So they lose MAT1A quickly, and SAMe level falls. This is work that was published long ago by Jose. So indeed, so we use this model to kind of recapitulate what's going on with the MAT1A knockout, if you will, because we know SAMe level falls. And indeed, in this model, you can see that DUPS1 mRNA level falls. If you put SAMe in the medium, you can protect it a little bit, okay? But look at the protein level of DUPS1 is almost gone completely, despite the fact there is still some mRNA level, okay? So the protein level is totally like down to 10% of baseline or lower, and that SAMe can protect against this loss. Almost not quite as good, but you know, compared to MG132 that you guys probably know is a proteasomal inhibitor. So this did a pretty good job. We then looked at human hepatocytes, and the same occurred. That SAMe was able to protect against the loss in DUPS1 protein, okay? Now, we had already suspected that SAMe must have an effect on proteasomal activity, and this further pushed us to investigate this further. So here we're looking at two measures of proteasomal activity, chymotrypsin-like and caspase-like activity, to indicate proteasomal activity. And you can see that upon culturing 12 hours later, there's an increase in proteasomal activity, and that SAMe can block that. Almost just as good as MG132, okay? Then she went ahead and purchased the semi-purified, we should say semi-purified because they're not really totally pure, 20S proteasomes. And using the 20S proteasome, she also was able to demonstrate that in an in vitro system, SAMe can inhibit the activity of proteasomes. Now, I'm not showing that data, but Manuel, who's I think in the audience, sitting somewhere, then showed that when you incubate purified proteasomes with SAMe, some of the subunits were degraded. Okay? So this is a very exciting area that SAMe can affect the stability of some of the subunits of these proteasomes, and then by doing that, control its activity. Okay? So what about the knockout mice? The proteasomal activity in the knockout mice? Well, here first, the SAMe level is lower in the knockout as expected, but we can correct this if you treat the mice with 1 week of SAMe. Okay, so you can normalize it. And here we see proteasomal activity in the knockout is higher, and that treatment with SAMe normalized it. And here's what happened to DUPS1 mRNA. It was normalized, and protein level completely normalized by SAMe. Okay, what's the mechanism of this mRNA normalization? Here, using chip analysis, what you're seeing is p53 binding to the DUPS1 promoter region that contains the element. And in the knockout, there's decreased binding, and this is normalized, tended to be normalized with SAMe treatment. So one of the mechanisms to explain the fall in mRNA of DUPS1 is decreased p53 binding to DUPS1. And we know it's upstream of DUPS1, and that SAMe can correct that partially. Okay? And as I showed you, ERK activity is heightened in the knockout mice. SAMe treatment normalized it. That higher DUPS1 phosphorylation that correlates with proteasomal degradation also normalized with SAMe treatment. So one of the key mechanisms leading to malignant degeneration is SAMI's ability to modulate proteasomal activity. And in this case, the target is DUPS1, okay? Degrading DUPS1, allowing for ERK activity to go unchecked, all right? All right, the other has to do with prohibitin 1. Now, many years ago, actually, the paper was published in 2003 in PNAS that was led by Jose and Fernando Corrales. Using proteomics, they identified prohibitin 1, this protein, not mRNA, okay, mRNA was unchanged. The protein was downregulated from time of birth and it persisted up to 8 months, okay? They were interested in looking for something that was persistently abnormal in order to better understand what might be the reason for NASH development, okay? That was really the underlying aim. Now, so prohibitin 1 showed up and we had no idea what it was. So we had to do a lot of reading. And basically, prohibitin 1 is a fascinating protein. It is a product of a nuclear gene that is mostly targeted to the inner mitochondrial membrane, where it heterodimerizes with a homologous protein called prohibitin 2, and they act as chaperone-like proteins, okay? So they protect many of the other mitochondrial proteins. But it's more interesting than that. They're also found in the nucleus. In the nucleus, they have been shown to modulate the transcriptional activity of E2F and P53. In E2F, it's been shown to be a repressor. In P53, it's been shown to be an activator, okay? Now, this was originally cloned using this differential display in regenerating liver. These investigators found that after you do a partial hepatectomy, prohibitin 1 disappeared before DNA synthesis even started to increase. So they hypothesized that this must be a tumor suppressor, and that's why it's named prohibitin 1, okay? But subsequently, it became controversial whether or not it's a tumor suppressor. Why? Because it's often overexpressed in cancers. But this is actually a target of c-MYC, okay? And c-MYC is an oncogene. So whether or not the overexpression is kind of a secondary effect because of c-MYC, we don't know, all right? So its role in oncogenesis is controversial. That's all I will say. Now, people have been trying to understand its role, but if you delete it, it's lethal. Okay, so much of what we know about the functions of prohibitin 1 comes from yeast, studies performed in yeast and in culture, in mammalian cell cultures. And I will also say that in that work published in 2003, they also included specimens from OB mice and obese patients, and they had reduced hepatic prohibitin 1 protein level, okay? So many years ago, Jose persuaded me to embark on generating a liver-specific prohibitin 1 knockout. And somehow I agreed. And it took 6 years and the lives of many postdocs in the lab. And finally, it was accomplished. And Kwang-Soo Ko, who finally characterized the phenotype of this mouse model, now has a tenure-track position back in Korea. So I will just share with you the essential phenotypes. So there's a lot of fetal wastage. A lot of the embryos don't make it. And there's a lot of death early on, okay? At 3 weeks of age, many of them look horribly sick. And this is what their livers look like. Very necrotic, a lot of inflammation. There's metaplasia in the bile ducts and anisocytosis. For those of you that are not pathologists, anisocytosis means different nuclear sizes, okay? And here, at 14 weeks, you're seeing dysplastic nodules. By 3 weeks, if you stain it with OV6, this is a stain looking for progenitor cells. Many are positive. GSTP is another marker for preneoplastic lesion, it's positive. And by 38 weeks, this is alpha-fetoprotein that I told you is a marker we use to detect HCC, and it is also a marker for increased growth, and it's positive. Now, ALT is 10-fold higher in these 3-week-old mice. Alkaline phosphatase is higher. Bilirubin is 5-fold higher. They're really sick. They have severe liver injury. Okay? Now, since it's a mitochondrial chaperone, naturally we looked at the mitochondrial morphology. And this is using EM normals. These are normal mitochondria. And you can see in the prohibitin 1 knockout, the mitochondria is swollen. Some of them, the cristae is disappeared. Okay, so very, very sick mitochondria. And using 4-HNE as a marker for oxidative stress, they stay positive. Over from 3 weeks to 14 weeks, these mice develop increasing apoptosis, increasing growth. It's just a compensatory response to all these injury. That's to be expected. And very impressively, even at 3 weeks, there's already increased fibrosis. Okay? Perhaps even more striking is by 20 weeks. Well, actually I should say that, yes, by 20 weeks, all of the mice have nodules, multiple nodules. And by sometime between 35 and 46 weeks, more than a third of them have liver cancer. Okay? Here is showing normal wild type. This is one of the liver cancer. You can see 2 there. Here's one, here's another one. And multiple cancers, some in different stages, some a little more differentiated, while another part of the liver has really undifferentiated HCC. Multifocal HCC occurs in this knockout mouse model. Okay? Now, the argument can be, well, you have so much injury, so much inflammation, so development of cancer is a compensatory phenomenon that has been reported in other knockout mouse models. So what is so exciting about this? This is to be expected. That's what the reviewers said. Okay? So since prohibitin 1 has been reported to behave like a protein that influences the transcription activity of other proteins, we investigate whether or not it could regulate directly in an in vitro setting. So we use this murine hepatocyte cell line called AML12, which is a non-transform, well, it's a non-malignant, I should say, a hepatocyte cell line. You can't really see this at all, but you have to take my word for it that there's this green is prohibitin 1, so it's staining mostly in the cytosol and the mitochondria, but it's also present in the nucleus, okay, which you really can't pick out at all. If we silence it acutely, you can get really a significant drop in the mRNA level. The protein level, this is like 18 hours of treatment, fell only slightly, but we get an increase in BRDU, which is an indicator of growth. Now, we then look at, using chip analysis, the binding of that E2F, which is known to be repressed by prohibitin 1. Okay? So we're looking at E2F binding to cyclin D1, which was induced with this acute knockdown. So the cyclin D1 promoter region that contains E2F binding sites are these, are shown here. The one, they're all increased with the knockdown, but the one in particular is this region. So you can see a dramatic increase in E2F binding to the cyclin D1 promoter to turn on this gene. Okay? What about the converse? Overexpression of prohibitin 1 decreased BRDU slightly. So at least in hepatocytes, it appears to behave like a tumor suppressor. Okay? So that's another area that we're very interested in. Now I will take you to yet a new area for many of you, most of you except for Jose, who knows this already. So that has to do with simulation, Samian simulation. Okay? And this is work that just got accepted. So what on earth is simulation? You probably all know, but I wasn't all that familiar with it, so I had to really do a quick reading. So simulation is a post-translational modification that is very similar to ubiquitination. Okay? So it conjugates sumo to target proteins, but in contrast to ubiquitination, which usually takes these proteins to get degraded, simulation does the opposite. tends to stabilize protein. Sumolation also affects protein-protein interaction, and it also affects protein trafficking in the cell. Okay? Now, UBC9, or ubiquitin conjugating enzyme 9, is the only sumo E2 enzyme. Okay? So it is absolutely essential for sumolation to occur. So it catalyzes this sumo conjugation. Now, UBC9 is overexpressed in cancer, many cancers, lung cancer, ovarian, breast, melanoma, but it's never been studied in liver cancer. It's known that genotoxic stress induces sumolation. And we already knew that MAT1A knockout mice have genotoxic stress, and they develop cancer. So it was natural for Lauda to ask, what about sumolation? What about UBC9? Okay? And indeed, she finds that in liver cancer, just like in many other cancers, there's increased UBC9. Okay? So this is just maybe one of the things that occur with cancer. And if you look at UBC9 protein, it is also higher. Now notice how the protein level goes up even higher than the mRNA level. This is kind of the opposite of what I just told you about DUPS1, right? And APEX1, which go down in MAT1A knockout, if you recall. What about knockout? Well, the knockout mice get an increase in UBC9 mRNA level as well. SAMI treatment doesn't do anything. Okay? The protein of UBC9 is also up in the knockout. With SAMI treatment, it tended to be normalized. So now you're going to say, wait a minute, you just told me that proteasomal activity is high in the knockout, and that with SAMI treatment, it inhibits protein degradation. So how on earth can you be telling me that this protein goes up in the knockout and that SAMI treatment lowers it? This is totally the opposite of what we were thinking. Quite counterintuitive, if you will. So I'll tell you why later, okay? Now, this is just looking at protein sumolation using SUMO1 antibody. And this is the big substrate, RUNGAP1. Often this is what people use to indicate protein sumolation. And you can see the dramatic increase in protein sumolation, and that it can be brought down toward normal by SAMI. Now, since we know UBC9 is overexpressed in many cancers, we then went to 2 different liver cancer cells, as well as colon cancer cell and a breast cancer cell line that I'm not showing here. But everything is all the same. Everything that I'm showing you occurs in all 3 cancer cell types, I should say. mRNA level of UBC9 is not affected by either SAMI or MTA. And that MTA is that compound I told you that can be spontaneously derived from SAMI. So whenever you're treating in vitro with SAMI, you should be comparing it with MTA, because it may be the MTA that's doing it. Okay? But at the protein level, both can reduce it by nearly half, 40 to 50%. Okay? This is 24 hours of treatment. Now, so it can't be proteasomal activity, obviously, because we're observing the opposite. So we therefore had a hypothesis that maybe SAMI and MTA alters the post-translational modification of UBC9, and that somehow affects its protein stability. So what do we know about post-translational modification that can be affected by SAMI? Well, years ago, Heping in the lab had shown that SAMI and MTA treatment can affect cellular phosphorylation status in HepG2 and Q7 cells. Okay? The way they did it was they actually increased phosphatase 1 expression. So by increasing PP1 and activity, you dephosphorylated proteins. Okay? So when you treat with these 2, you generally get a dephosphorylation of proteins in cancer cell lines. And that was, in fact, true. So looking at overall phosphorylated proteins using anti-phospho antibody that would pick up all kinds of phosphorylated antibodies, you can see a generalized decrease in both Q7 and RKO cells. after treatment with SAMI or MTA. Now, UBC9 has never been reported to be phosphorylated. So Lauda immunoprecipitated with anti-UBC9, then blotted it with phosphoantibody, and lo and behold, it's actually phosphorylated at baseline. And with these treatments, it went down. Okay? Total UBC9 went in the same direction, and again, RON-GAP1, sumo RON-GAP1 as an indicator for protein accumulation, inhibited by these treatments. Okay, now this would be suggestive that when UBC9 is dephosphorylated, it's unstable. What about if you were to block dephosphorylation? Okay, so she treated the cells with a pan-phosphatase inhibitor, a mixture. So all the phosphatases were inhibited. By doing that, IP-UBC9 blot for phospho-UBC9 using anti-phosphoantibody, we see an increase, about 27% of that level of phosphorylated UBC9, and the total UBC9 also went up in parallel. So this is, again, very suggestive that the status of phosphorylation of UBC9 can alter its stability. So we went to the sequence. So this is the sequence of UBC9. And she used several different websites and what have you. But the bottom line is that using all of these tools, she identified several sites that are potential sites for kinases to phosphorylate. And one in particular, this right here, these double serine, serine 70 and 71, interestingly, that had been reported long ago in yeast, that if you mutate that, the protein was unstable. Okay, so that was very interesting. And right here, one of the kinases immediately piqued our interest, and that's CDC2. CDC2 had a really high score, making it very highly likely that it can phosphorylate this protein. In addition, CDC2 is overexpressed in cancer, okay, making it particularly interesting. So you can purchase recombinant CDC2. You can also purchase recombinant UBC9, okay? So that made it very easy for Lauda to demonstrate that in a test tube, CDC2 can phosphorylate UBC9. And this can be inhibited using roscovitin, which is an inhibitor of CDC2, okay? Now, we then went to the knockout animal specimens, and here, IP-UBC9, again, we see the increase in phospho-UBC9, and this can be lower with SAMI. Total level, I showed you before, high and lower with SAMI. And here is the first demonstration in vivo that the two interact, okay? So IP-UBC9, you bring down CDC2. So there's increased interaction in the knockout mice livers, and this can be inhibited by SAMI, okay? Now, since CDC2 seems to be the kinase that's doing the job, so we looked at how these compounds, SAMI and MTA, might influence CDC2 expression. And interestingly, they inhibit CDC2 by about half. And this is occurring at the mRNA level, okay? And protein level is the same. Now, to understand, is this the mechanism of how SAMI and MTA lowers UBC9 phosphorylation? Here, we're knocking down CDC2, or treat it with SAMI or MTA alone, or we combine them, okay? So you can see knockdown of CDC2 lowers UBC9 total level by about half, almost the same as SAMI and MTA, and combining them did not give you any additional inhibition, lowering, I should say, okay? And this is strong evidence that CDC2 is the mechanism by which SAMI and MTA inhibit or lowers UBC9 phosphorylation and lowers its stability. Now, in the tumors, both liver cancer, colon cancer, and also, I'm not showing you, breast cancer, The tumors themselves have higher phospho-UBC9 levels, total UBC9, okay? Total CDC2 and CDC2 that is bound to UBC9, okay? So this is also the first demonstration in cancer specimens. Now finally, what about the proof that those serine residues, the targets of CDC2 is really that important? So Lauda went and developed expression vectors of UBC9 that's either wild type or mutated in the serine 70 or serine 71, okay? Using an HA tag. So here we see IP using anti-HA. Here we see the protein level when you have the mutant at the 71 serine position is markedly reduced, but not the one at 70, okay? Interaction with CDC2 is also lost by half or more when you mutate the serine 71, okay? And UBC9 total level, after IPing with HA, this recombinant one is also lowered. What about protein half-life? So here, either wild type or the mutant 71 was expressed, and the half-life of protein was measured after inhibiting protein synthesis with cyclohexamide, and you can clearly see the mutant is far more unstable than the wild type, okay? So this is the complete story. SAMI-MTA can inhibit CDC2 expression, which then lowers the level of UBC9 phosphorylation at, in particular, serine 71, leading to a more unstable protein. This is her cartoon to summarize her findings. Under physiological conditions, you have a certain amount of CDC2 phosphorylating UBC9, and sumylation is occurring to target proteins. This is all physiological. But in cancer, or genotoxic stress, oxidative stress, you get an increase in UBC9. There's some report of some sumo going up as well. CDC2 goes up, increased phosphorylation is occurring, increased sumylation is occurring because there's more UBC9 because it's more stable. A barren sumylation. How that leads to cell growth, that's lots of question mark. But with SAMI-MTA, if you treat using these compounds, you're blocking a very early step that is required for all these barren sumylation to occur. Okay? All right, changing to a different subject. So far, I've only told you about MAT1A and how important it is, that if you don't have it, you get all these pathways going crazy, and they all lead to cancer. Okay? The other two MAT genes are actually really fascinating and equally important as well. I just don't have time to tell you. You'll have to keep me here another day to finish all that story. Anyway, why are they so important? Because they're both induced in liver cancer, and they're both seem to facilitate cancer growth. Okay? Now, in liver cancer cells, Komau in the lab has shown that leptin is mitogenic, but it requires the MAT2A, MAT2B induction. Okay? And interestingly, this MAT2B is fascinating. It doesn't just encode for the beta subunit to regulate MAT2. It regulates many signal transduction pathways. Okay? And it apparently also regulates STAT3 activation by mechanisms that have yet to be uncovered. In stellate cell activation, Komau has also shown that these two genes have to be induced. If you block their induction, stellate cells are not activated. This is the area of her current research program now, understanding the mechanism of their induction and how targeting them may be a way to treat fibrosis. Okay? Now, in stellate cells, and actually also in liver cancer cells, MAT2B also control ERK and AKT activity, again by mechanisms that we're investigating. Okay? So this is just to kind of tell you that they're very, very interesting. And I will tell you a little bit more about MAT2B because I know that Adriana, are you here? There you are, yes. Okay. All right, so Adriana is working hard on this project. So MAT2B. Now, all the published work in the past that talk about this beta subunit is one of the variants of MAT2B, okay? Now, Heping in the lab uncovered that there are multiple isoforms of MAT2B. Variant 1 and variant 2 are the two dominant variants. They are different in the N-terminal by about 20 amino acids. The variant 2 uses this 2.3 kb upstream region to the transcriptional start site. So this is different, but everything else from exon 2 onward are identical. Okay? So variant 1 is the one that's known to regulate MAT2. Whether or not variant 2 also regulate MAT2, we don't know. When we look at expression of these variants, we find that they're differentially expressed. So in other words, like some tissues like the bone marrow, trachea, adult liver, placenta, using northern blot, you don't see them. Other tissues predominantly express variant 1, some mostly variant 2, like skeletal muscle and testes and heart, okay? So we knew from Malu and Jose's work that MAT2B is induced in liver cancer. But we didn't know which variant, because at that time we didn't know there were variants. So you can see that both are induced, okay? Probably variant 2 is induced even more. Now, do both variants regulate growth? Yes, they do. So using HepG2, which expresses both variants, okay? Although it expresses more variant 1 than variant 2. So if you do the knockdown, and the knockdown efficiency is comparable, you can see that there is more MAT2B left following V2 knockdown. And that's just illustrating there's more V1 in HepG2. There's decrease in DNA synthesis, okay? If you overexpress, and overexpression, we usually typically use HU7. HU7 cells express very low level of MAT2B. So overexpressing using HU7, you get an increase in DNA synthesis. Okay? So both of them regulate growth. I don't have time to show you, but I can tell you both of them, in particular V1, regulates apoptosis. Okay? So we took a proteomic approach to understand what are the proteins that interact with these variants. Because they are very interesting. They must be doing stuff that we don't know about. So let's use an exploratory approach. So Mengsha in the lab at that time developed bacterially expressed histag proteins, V1 or V2, use them to do a pull-down assay. Okay? So a lot of the proteins could be purified, pulling down, mixing it with cytosol, and this using in-solution proteomics, we brought up a number of proteins that interact with both variants, only variant 1 or only variant 2. So if you look at some of these proteins, well, it's very nice that MAT2B came down. That's reassuring, okay? So we expected that. Now, there's splicing factor, pre-mRNA. Many of these are in the nucleus, okay? And then there's this one, Malu's favorite, one of her favorite, HER, also showed up on the list. So we knew that HER was important in growth, okay? But look, there's also stem cell growth factor precursor, and there's a G protein, GIT1. This is an area that we're investigating right now in the lab. So let me tell you about HER, because this was published. So, so she then verified that indeed there's HER present, and indeed in the pull-down assay, there's a V1 or V2, you can pick up HER. And if you immunoprecipitate HU7 cells overexpressing either V1 or V2, okay, and the expression vectors is V5 tagged. So using anti-V5, you can bring down HER, okay? So this is recombinant. And endogenously, precipitate using anti-HER, you bring up MAT2B, anti-HER, MAT2B in both RKO and HepG2 cells, okay? And the reverse is also true. You can immunoprecipitate in both directions. All right, so where is it? Since there are so many proteins are nuclear, so she then looked at cytosol versus nucleus, in all these cell types, and she finds that most of MAT2B is nuclear. Okay? And this is true for endogenous proteins, and it's also true when you overexpress V1 or V2. Okay? Tends to be found in the nucleus. And this is just using confocal. Green is now staining for the recombinant protein that it's nuclear, predominantly. All right. Now, if you overexpress V1 or V2, what happens is that her cytoplasmic content goes up. Conversely, if you knock down V1 or V2, okay, cytoplasmic her goes down. So depending on the level of expression of MAT2B, either variant, you can affect the her intracellular localization. And this is demonstrated using confocal with V1 overexpression. You see her now staining in green, more of it appearing in the cytoplasm. Same goes for V2, okay? And indeed, if you overexpress V1 or V2, you get an increase in her targets, cyclin D1, cyclin A. And if you were to knock down V1 or V2 knockdown, her is unchanged, but cyclin A, cyclin D1 goes down, as does growth. Okay? So one of the mechanisms that MAT2B controls growth is by this mechanism. And there are others that we're working on, obviously. Okay, so let me change to talk about chemoprevention. Okay, so it's been known for a long time that SAMe will prevent the development of liver cancer in animal models of liver cancer. The mechanisms, there are multiple mechanisms that include preventing hypomethylation of proto-oncogene promoter regions. It includes selective pro-apoptotic effect of SAMe on cancer cells. And we have also shown that SAMe exerts an anti-angiogenic effect. Okay? Now, however, I can tell you that using an orthotopic model of liver cancer, SAMe was not effective if you already had liver cancer present. And that's because long-term treatment of SAMe led to a compensatory increase in GNMT expression that removed excess SAMe and prevented its accumulation. So if you treat the animal for 1 day, you can drive up the liver SAMe level by 10-fold, but if you treat it for 3 weeks, it only goes up by about 30%, okay? Because of the methyltransferases are getting turned on and eliminating SAMe, so it cannot build up. All right. We then tested this idea, if you force MAT1A expression, would you have a more stable SAMe level? Indeed, you can. The liver cancer cells overexpressing MAT1A had a doubling of intracellular SAMe that was stable in vitro and in vivo in the tumors. By doing that, they exhibited reduced growth, okay, and reduced angiogenesis, ERK, and AKT. All right, well, this is all fine, but it's not practical. You can't take liver cancer and try to transfect MAT1A. So you have to have a better way of targeting. So what's another way that we can bring up MAT1A expression? Well, it's been reported that methylation is important in the promoter, but that's difficult to manipulate, okay, in that setting. So we had the idea, let's look at microRNAs. So as probably all of you know, microRNA is a hot field. Family of small 21, 25 nucleotides, small RNAs that generally, and I mean generally because exceptions to the rule have been reported many times now, but they generally negatively regulate gene expression. In humans, they make up more than 1% of the gene repertoire. They regulate more than 30% of protein coding regions. As of recently, close to 2,000 microRNAs have now been annotated, and most have not been characterized, okay? We know very well that in cancer, there's dysregulation of microRNA, and it is so important. Why? Because they serve as wonderful biomarkers, okay, because they're found in the blood. not only biomarkers, but they're great therapeutic targets. They're so small, it's very easy to deliver different ways to manipulate their levels. If they're downregulated, you can increase their expression. If they're overexpressed, you can downregulate it. This concept in treating liver cancer was published in a Cell paper a few years ago. A beautiful work that showed one injection, just one, via the tail vein, was able to have a sustained effect on the microRNA expression and on tumor growth. So that was very exciting, okay? So this is a really very practical and achievable aim to target microRNAs in human HCC treatment, and human cancer treatment, I should say. What I'm going to show you in the following few slides are unpublished data, okay? So we asked, do microRNAs regulate MAT1A, okay? All right, so using well-known websites, Target Scan, you name it, we had a long list of potential microRNAs, okay? So there were some that's already been reported in HCC to be abnormal. We weren't interested in them. We wanted to look at those that have not been reported. So we only looked at those not reported, those with high scores, preferably those whose functions are not known, okay? And we came down to these three. Since we started the work over a year ago, I will tell you that there is one paper that showed in breast cancer that mere 495 is important, okay? But none of these three have been reported in HCC. Okay, so we took our HCC specimens and looked at some candidate microRNAs. So these are the top 5 in terms of score and unknown roles in HCC. So 2 of them are actually downregulated. So we didn't want to look at those because we have to focus. So we looked at 3 that are increased in HCC. Again, why? Because generally they downregulate expression of genes and proteins. And we know that MAT1A is downregulated. So we're looking for microRNAs that are upregulated, okay? So that's why we concentrated on these 3. So here is using 2 different liver cancer cells, HepG2, Hep3B, that knocking down these microRNAs, either 664, 4853P, 495 individually or in combination, raise MAT1A mRNA level and protein level in both cell types, okay? If you go to the 3' UTR MAT1A, here are the binding sites for these microRNAs. There are 2 for mere 495, 1 for the other 2, okay? We then develop constructs that's either wild type or mutated for these binding sites. And here is the effect of inserting a wild type 3' UTR to a reporter construct, or those that contain mutations. With the wild type, you can see this dramatic fall in reporter activity, which confirms that these are in fact functional, because when you put it in a reporter system, it's inhibited. It's inhibiting the luciferase, okay? But if you mutate these sites, particularly when you mutate all the sites, you almost recover all of the reporter activity. So this confirms that these sites are functional and that these microRNAs regulate MAT1A negatively at the mRNA level, okay? All right, so that's all fine and dandy. Let's go in vivo. So here, Heping then developed cell lines that would stably express overexpression of these individual microRNAs. And stable expression of these microRNAs brought down MAT1 expression. Stable expression of the siRNA that target these microRNAs raised MAT1A level, okay? So these cell lines were then used to inject into nude mice. And here on the left is forced expression, on the right, knockdown. So forcing the expression of these microRNAs, which lower MAT1A further, you can see how big these tumors became, okay? And you can see this is the control, the increase in pCNA staining, and the fall in MAT1A immunohistochemistry in the tumors, okay? The converse, knocking it out, this is the scramble control, knockdown of these microRNAs raise MAT1A expression, reduce the size of these tumors, lower the pCNA staining, raise the MAT1A immunohistochemistry. Okay? So that's proof of concept in a new mouse model. But we wanted to know a little bit more. So here what we did was we took HepG2 cells that are capable of invading, okay? So we injected the HepG2 cells into the left lobe of the liver. Then we waited up to 8 weeks, okay? Then they were treated, the animals were treated with siRNA against this mirror, or that mirror. Every 2 weeks, times 0, and then every 2 weeks, okay? This is at the end of the 8th week, at the site of this injection in scramble, that's the tumor volume, and these are the treated, okay? This is N of 8. But what's really exciting is that when you look at tumor invasion and metastases, the scramble treated had involvement of the diaphragm, half of them lung, all of them had involvement of the abdominal wall. You can see the 495, none of them had metastases to the lungs, okay? Diaphragm, and only one, abdominal wall, okay? So knocking down these microRNAs impacted on invasion and metastases and tumor growth, okay? Obviously, I'm not saying MAT1A is the only way they're doing it, because clearly, while all three can affect MAT1A expression equally, miR-495 was really potent, okay? So miR-495 have other targets besides MAT1A. But nevertheless, these two was also effective. So that shows that MAT1A does play an important role. So how can you change tumorigenicity, invasion, and metastases just by changing MAT1A expression? How can that be? Well, what is MAT1A? MAT1A encodes for this enzyme that makes SAMe. What is SAMe? Methyl donor. It's a methyl donor, right? So we decided to look at methylation. So here, these are the cells that were stably expressing siRNA against these microRNAs. Okay? Here we're looking at overall global DNA methylation. And when you knock down these microRNA, you raise MAT1A, global methylation was up. Conversely, if you overexpress these microRNAs, MAT1A goes further down, and you get a hypomethylation. Okay? Very exciting data is that when you use confocal, when you look for MAT1A, when you overexpress, I should say not overexpress MAT1A, when you knock down these microRNAs so that you get an overexpression, an induction now of MAT1A, it tends to go to the nucleus. Okay? So you can see in the control as compared to the siRNA treated, how bright these nucleus now look. So MAT1A is in the nucleus, and that's been already reported by another group, so that's not novel. Okay? But in the nucleus, it can change methylation of a lot of genes. We have already identified one gene to be particularly important because that gene's been recently very hot and well described to play an important role in invasion and metastases. And indeed, when we knock it down, this gene becomes hypermethylated, and when you overexpress, the opposite occurs. Okay? So there's obviously a lot of targets that this is going to influence. So this is just the beginning of uncovering how that might work. So let me then end by telling you that I have shared with you MAT1A deficiency will result in abnormalities of a lot of signal transduction pathways in liver cancer. In addition to being a methyl donor, it regulates proteasomal activity and sumylation. MAT2B variants regulate growth. One of the mechanisms is through influencing her intracellular localization. And SAMe is effective in chemo prevention, but enhancing MAT1A expression is a better strategy for HCC treatment. Okay? And finally, it's the same picture that I show over and over. I got to get new pictures. That's Lauda. A lot of her work I showed you today, that's Meng, and that's Kwang-Suk, who characterized the prohibitin 1 knockout, and that's Heping, who did a lot of the work that I showed you. And all the microRNA work are really started by Heping. And of course, I don't have to tell you, Jose, my buddy, Malu, wonderful friends here, and all the collaborators around the world, and of course, NIH money, without which none of this is possible. So I'll stop here. I am so sorry I went over time. I didn't think it would be that long. I'm so sorry.
Any question?
Shelly, you've mentioned that MTA is a degradation product of SAMe, right? And in some way it can mimic the role made by SAMe in some of those pathways, obviously not those involving methylation. But have you checked whether that can be extended also, I mean, regarding treatment with MTA, to other molecules such as S-adenosylmethionine or even adenosine itself? For example, SAH is closer structurally to SAMe than MTA, right?
Is toxic.
It is because of toxicity, okay.
SAH is very toxic. It induces apoptosis, ER stress. SAH is also a potent inhibitor of transmethylation reactions, so it is not something you want to use. You want to keep it as low as possible.
And what about adenosine, for example?
Adenosine's got so many effects, I wouldn't go there.
Oh.
Yeah.
No, I have a few questions. These variants of MAT2B that you show that they are regulated during the proliferation, what happened during liver regeneration, if you know something, or could be there under the control of AGF or MP kinase?
You mean MAT2B?
Yes, MAT2B variant. They
yes,
move in response to growth
HGF will
factors?
induce MAT2B.
Sorry?
HGF induces MAT2 beta.
Okay.
Yeah, both variants are induced. We've not published that yet, but yes.
And they move also. That AGF move the location of them?
That we've not looked at. Yeah. I think there's a question in the back there. Yes?
Sorry, one quick question. This effect of SAM on the proteasome, has it been shown in tissues or cells other than liver? The effects of SAMe on the...
Only liver.
On liver.
Mm-hmm.
Hi, Shelly. Thanks for your nice presentation. I have a question about your prohibiting the knockout mice. I mean, I think the phenotype is just amazing. So I have two questions.
I wish the reviewers would agree with that.
I mean, they look amazing to me. So I have two questions regarding inflammation. So one, you think that just blocking, I mean, of course, first characterizing inflammation, who's really doing the damage, and blocking the inflammation, do you think that will reverse the phenotype, or you think just blocking prohibiting in the hepatocyte just does the job, and then you go to an HCC?
Okay. The inflammation is a response to hepatocyte cell death, because the mitochondria, the deletion is only in hepatocytes. So the abnormal mitochondria is only occurring in hepatocytes, or whatever cell that's expressing albumin, because it's an albumin creep. So I don't know what contribution the inflammatory cell, the inflammatory response has to the later development of fibrosis and cancer. That I cannot address. But there's a lot of inflammation. So that would be something that you can look at, in addition to the other fight project that you're already looking at. Yeah.
No, the thing, because my second question goes in that direction. I mean, you have a hepatocyte-specific prohibiting, but prohibiting is expressed in every cell,
ubiquitous,
right?
Yes.
Yeah, so, I mean, there are many tools out there. You can have a Liskray mouse that just blocks prohibiting in mieloid cells. You have an MXCray, you block it everywhere. Because, I mean, we know from other factors like NF-kappa B that if you block it in a hepatocyte, that drives the liver into a state of hepatitis. But if you block it in inflammatory cells, you are protected against. So I know it's not a transcription factor, but you say that prohibiting regulates other proteins. And I mean, and the mitochondria is very important for the cells. So do you think it would be worth it to block prohibiting in those non-parenchymal cells? Do you think that would have a different effect than just blocking, prohibiting the hepatocytes?
I don't know. It's speculative at this point. I think another area where prohibition is very interesting, I think, is the colon, okay? Because it's been shown. I should also tell you, prohibition is shed into the blood, so it's found in circulation, okay? What it's doing there, who knows? What is it binding to? You know, don't know. There's been some noise that it may play an antibacterial role, okay, especially in the gut epithelium. Now, the only thing that's been published prior to our knockout in the liver is transgenic mice that overexpress prohibitin in the colon. And these mice are protected from chemical-induced IBD, okay, chemical-induced colitis. And the same group now has work that's under review that suggests it may have a role in colon cancer as well. So that's interesting. And in the gut, it seems to enhance the transcriptional activity of NERV2. NERV2, which is an important transcriptional factor that binds to antioxidant response element. And they speculated that that may be the reason why, when you overexpress prohibitin 1, that there's less injury, because it's enhancing the antioxidant capacity. Yeah.
No more questions? Thank you very much, Shelly. It's been a pleasure.
Thank you.